Hello, everybody, and welcome to Recorded Asynchronous Lecture 4 for Bio 145 Microbiology. This lecture will be about bacterial structure and growth and nutrition. It's kind of part 2 of that. And this lecture is scheduled for Wednesday, January 21st, 2026. In terms of announcements, there's no announcements. From now on, I'm just going to put announcements in the live lecture PowerPoints, all right? Because it gets kind of repetitious and time consuming to put them in both. So from now on, announcements are only in the live lecture ones. Okie doke. Well, let's get going then. All right, before we go into some new stuff, just recapping, we're going through some basic structures that are found in pretty much all bacterial cells, eubacteria, eubacteria. So before we talked a little bit about plasmids, ribosomes, we used to make proteins. We talked about things in the cytoplasm, plasma membrane or cell membrane, and we spent a lot of time on cell wall, gram positive versus gram negative. Now we're going to go on to things like outside the cell wall. We're going to talk about the glycocalyx, which you can see here, the glycocalyx right here. And then we're going to talk also about pili right here. All right, and some flagella. All right, and I'll just say these things are optional equipment. They're found on many, but not all bacteria. So we'll start with glycocalyx and go on from there. And just a note, this recorded lecture was used for my Tuesday, Thursday lectures, okay, in the past. So at the beginning it says, this is lecture blah, blah, blah. Don't worry about it being lecture 3 or whatever. You're on lecture 4. I just don't want to do re-record the whole thing over again. So let's get talking about glycocalyces. Hi and welcome back. I'm your host Jeff Lynn Cheragino, and this is part 2 of Asynchronous Lecture 3. So now let's talk about some more structures, and we've started inside and we're going outward. So let's go outside now, outside of the cell wall. So we're thinking this thing right here, which you can see. It's on the outside of the cell wall, and it's found in some, but not all bacteria. And it has a lot of medical importance, and it's called the glycocalyx. The glycocalyx is a combination of proteins and polysaccharides. It's kind of like peanut butter. It's gooky, it's ooky, and it's on the outside of the cell wall. And there's really two types of glycocalyx. You call a glycocalyx a capsule if it's tight and kind of hard to wash off. It's kind of thinner. On the other hand, you call it a slime layer, like you'd think it's slimy. Usually if it's thicker and broader and much more softer, gooky looking, all right? But they're all synonymous with glycocalyx. Now, here's an interesting thing, and this is why we focus on it a lot in biology. If you have two strains of the same species, so they're pretty much identical, and the only difference is a few genes that help create proteins for the glycocalyx. I can tell you that the one that cannot make the glycocalyx, everything being equal, the one that cannot make the glycocalyx will be less pathogenic, meaning it will be less likely to cause disease, to cause damage to your body. Well, why? So why does the glycocalyx enable a bacterium to cause more damage in your body? And therefore, if it doesn't have one, it's less harmful. Well, we kind of have to look at what a glycocalyx does to understand that. So let's look at this picture. Maybe we get some idea. On the left, we have the glycocalyx type known as a capsule. All right, and it's kind of shown here, and it's kind of thin. All right, it's kind of thin, not that wide. It's outside the cell wall. And here is the slime layer. And look, slime layers are much wider, gookier, softer. So you think things on the outside. Well, what do you need things on the outside of? Well, and think peanut butter. Well, peanut butter's sticky. Yeah. Peanut butter's a coating. I mean, if you coat yourself with peanut butter, you'd probably maybe sweat less. Yeah. What else have you ever rubbed peanut butter all over your body? Okay, this is a family show, so I ain't going to go any more like that. But just think of that, putting these coats of sticky, gunky stuff on the surface. Well, let's look what it does to the bacterium. Number one, it prevents desiccation. So it's less likely to lose water and dry out. Makes it a little hardier. Okay. Tougher. Number two, it's a shield. It's kind of like something that will kind of stand between the cell wall and whatever's outside. Maybe poisons, maybe acids, bases, things that can hurt the cell. Well, that glycocalyx is kind of like a protective covering, a shield. Number three, it's protection specifically against phagocytosis by human cells. Now, I think a lot of you have heard this before, that in your body you have phagocytes. They're good immune cells, and they're kind of like Pac-Man if you're into retro games. They're just a little thing that kind of swallows up stuff, phagocytes. Now, you can imagine it could have a bad bacterium, and the human phagocyte wants to swallow it up and kill it. But imagine if it was covered by peanut butter. Would it be as easy to swallow and chew up and break down? I mean, cover peanut butter in something and try to swallow it whole. You can't. Well, this peanut butter-like covering of a glycocalyx also makes it harder for phagocytes, human phagocytes, to swallow up and break down bacteria if they're covered with a glycocalyx. So you can see maybe 3 reasons why. Let's go to the 4th reason why these things are bacteria with glycocalyces are more difficult to kill and more likely to be damaging and pathogenic. And this is the big one. This is the 4th one, is adhesion. Okay, it's sticky. Peanut butter sticks, almost like a glue, right? If you couldn't buy Elmer's. So the bacteria uses the dextrans. Okay, these are just polymers of glucose that are found in the glycocalyx, and they use them to form biofilms. So literally what they're doing is that dextrans, those polymers of glucose, are acting like a glue to attach one bacterial cell with a glycocalyx to another bacterial cell to another bacterial cell. And also they use that glue to them stick as this big mass of bunches of cells. They'll stick to each other, and they could stick to some surface. Okay, so they're sticking to each other and to a surface. And then you got a lot of other stuff. Once you've got that in there, other things can stick in there too. And altogether, that's known as a biofilm. All right, these layers of bacteria that are stuck to each other and maybe stuck to a surface and have stuff caught in them. It's a biofilm. Well, I'm sure you've encountered a lot of biofilms. If you ever had to clean some pipes in your house, there's got this gooky kind of stuff in there, slimy. Okay, slime layer, biofilm. If you have a pet, you don't change their water dish in a few days, there's some stuff growing at the bottom. It's sticky and icky looking. That's a biofilm. But stuff that I know you have right now in your body is plaque on your teeth. You eat a sugary meal or something like that, even right after brushing, and within hardly any time, suddenly you got this film, this slimy like film, and that's a biofilm. That's the bacteria forming, using their glycocalyces to begin with, to form a biofilm on your teeth. Now, if you watch this video, unfortunately, like I said, I can't play it for you, but if you watch this video, please put it into your browser and watch it. And it just shows really quickly the formation of a biofilm on the teeth, and known as plaque. So a person brushes their teeth, they eat some sugary stuff, and it's a time thing, and you can see the plaque forming like within an hour. It's like so much biofilm, so many bacteria forming there. So like on this picture here, I like this picture. So you see the plaque. If you were to look under a microscope, that plaque is just a bunch of bacteria stuck to each other, stuck to the surface. And the glycocalyx is that gluey material. Here's a really interesting cocktail party conversation. And I even wrote it out because it's an amazing thing. And when I started teaching micro 20 years ago, this was not known. The research on glycocalyces and biofilms are fairly recent, and it was not understood, but now it's understood. Here's amazing: 70% of bacterial diseases in industrialized nations, diseases of the respiratory tract, the heart, urinary tract, it's associated with bacteria that form biofilms. So biofilms are really important in helping bacteria make you sick. Okay. Another means of attachment used by many bacteria, though not all, but some, is pili. Okay, pili are also known as fimbriae. These are a long, thin structures. Let me show you this in a second here. And they project out from the plasma membrane. You can kind of see them here. Okay, they're coming out here, these long, little thin-like hairs. And they're mainly used for attachment. Okay, they attach bacteria to bacteria, or bacteria to bind a host, or bacteria just bind surfaces. And so they are also involved in biofilm formation. So if you see a bacteria making a biofilm, such as plaque, it could be glycocalyces, it could be pili, but often it's both. Usually bacteria kind of has both. All right. Now, one special type of pilus, one pilus, many pili, and its attachment, but attachment for a special reason, are called sex pili. And they're shown here. There's one sex pilus between one bacterium and another. And what it is this pilus is just kind of hollow. And it allows for DNA to be transferred from one bacterium, all right, here's the donor, to another bacterium, which we'll consider the recipient here. All right, so the DNA goes through this pilus and into the recipient bacteria. And so you kind of like to think of it's like they grab each other with loving arms, and these arms are like tunnels, and they send DNA over it. All right. And also, a final thing is one function for pili is sometimes they give a twitching motion, and they're actually used for locomotion. All right, they're used for locomotion. So when you see pili, you want to think attachment, helping form biofilms. You want to think, oh yeah, it's a way of sending DNA from a donor to recipient, and you also want to think a way of moving. All right, so there's many purposes for a given pilus. I know a lot of you grew up, when my kids grew up, and one of their favorite video characters was Larry Boy, and Larry Boy was part of the Veggie Tales, a group, and Larry Boy was a cucumber. Okay, you might some of you know this. Oh, come on, you're by yourselves. You can admit it. Is a cucumber, and a cucumber has no arms or legs, so he always had visions of being a superhero. So he was Larry Boy the superhero, and when he attached things, he used plungers. Okay, that's what he used for arms. All right, and you think these plungers were kind of like pili, if you want to think about it. Okay, they're extensions, and they use it for attachment. So Larry Boy used plungers, kind of like bacteria use pili or fimbriae. Long, thin attachment. Pretty good analogy, I think.
So to really appreciate what pili can do, remember pili are made of a protein called pillin. So the pillin gene encodes the pillin protein, and that gives you pili. We're going to tell you a story in live synchronous lecture about an experiment where volunteers were either infected with bacteria that had a functional pillin gene, or those bacteria that had a mutated non-functional pillin gene. And it kind of proved the importance of pili. So give you something to look forward to when you come see me in person. All right.
All right. The next thing is sometimes that's mistaken for a pilus or pili, but it's totally different. That's flagella, and flagella are used for motion. They're used for locomotion. So here's just your picture of kind of how it sets up, and you're kind of looking at it. Kind of almost has like a motor to it. Don't worry about all the parts here. And here's the flagella, okay? And it's mainly composed of a protein called flagellin, all right? That's what it mainly is. And its movement is stimulated by chemotaxis. So a bacteria can sense a chemical, and if they can either go toward it because of chemotaxis, so they're attracted, or they repelled, so they want to go away from it. So how can they go one way and go the other? Well, pretty amazing. They just do it with their flagella, whether they have one or two or many. And it's basically direction. So the bacterial flagellum, the bacterial one, is like a propeller on a train, on a plane. Sorry, propeller on a plane, and it can go up to 100,000 RPM. So it's pretty amazing. You get the idea. And pretty much whether it goes clockwise or counterclockwise or forward or reverse, that will dictate whether the bacterium goes forward or reverse. So you could imagine, just as an example here, so the bacterium is moving here on a run here. You could say, let's say it's spinning its flagellum like a propeller clockwise, forward. And now it's going to go forward. All right, and here it's going forward here. All right, but let's just say it was going to go backwards. Let's say it was going to go backwards. Let's say it wanted to go backwards. Then it's used the flagella, but it would go counterclockwise. Okay, so the direction of the spin, counterclockwise, clockwise, or forward or reverse, that would dictate whether the bacterium goes forward or backwards. A pretty simple system to do some fairly complex movement, as you can see here. So it's pretty neat what the bacteria can do there with just their simple flagellum. And the flagellum, again, it's got this long filament, and it's got, basically this is kind of like the motor or the engine for it. And you see, what I want you to notice, it has to go through a lot of layers. And right now we're looking at was that a gram positive or gram negative? It's got an outer membrane. Oh yeah, that's a gram negative, right? It's a gram negative bacteria. Gram negative bacteria. So it's a G minus. I'll put there. You can see it. So that engine of the flagellum's got to extend through all those different layers. Now, on the other hand, if you have a eukaryotic flagellum, let's say on a protozoan, it's going to work not like a propeller. It's going to work like a whip. Like a whip. And you know what whips look like. And it's made of the protein called tubulin. So if I have a mystery microbe question for you, and I say, oh, it's got a whip-like flagellum, that's a hint that that's a eukaryote. If I say it's a propeller-like flagellum, then you got to think, oh, that's got to be a bacterium. So that's important. Now, flagellum, this kind of brings out, just a simple point I wanted to make. The flagellum has these parts, and they have to extend, again, through all the layers, okay? Whatever the plasma membrane, if there's an outer membrane, whatever. And all I'm going to show you here is, here's a gram positive flagellum, here's a gram negative flagellum. That's the structure. And you can see they're different because this one's got to go through several different layers. This one doesn't have to go through as many different layers. So the properties of the gram positive versus gram negative cell wall, those will influence flagellar structure. So cell wall is not just cell wall. The cell wall affects a lot of things about the cell. In this example, it is affecting how the flagellum is designed in that bacterium. That's all I want you to know. Don't worry about all the names here. Just know the idea that the structure of the cell wall affects the structure of the flagellum. Cell wall is very important. Now, of course, there's got to be fancy scientific names, and you got to know these. And they're used to describe bacteria, and the names are based on the location and number of flagella. So the first one, if you have bacterium has one flagellum, it's monotrichous. That's not hard. Mono for one. Trich, that's when you're starting to see something like a flagellum. Amphitrichous means it has one or more flagella at both ends. Ambidextrous, so that's the guy there. Two. So it's at both ends. One or more at both ends. Lophotrichous means you got a lot of flagella, but they're only at one end. So here's lophotrichous. I think a lophotrichous is like saying you have a tuft. A tuft of flagella at one end only. And finally, some bacteria they're covered all the way around. They're think of perimeter, all around the perimeter of the cell. So we say they're peritrichous. They're covered with flagella. And this reminds me of the story of where it's my brother-in-law Tony. He has a friend named Wes. And Wes, the nicest guy in the world. But Wes, he's one of these people, have you ever noticed, he's really hairy, He wears a t-shirt, but like you could see the hair kind of creeping out from under his chest, over the collar of his shirt, or out from the ends of his of the sleeves. It's like the hair is just coming out, trying to invade out. They're just covered. He just was covered with hair. And I used to call him Perry. And he's like, why do you call me Perry? And I said, oh, just something. It was because he made me think of Perry Tricus. Yeah. What do you? Yeah, he kind of looked like that. Okay, you get one of those people. All right. So imagine if this wasn't a human being, it was a bacterium. That's kind of what the surface of a Perry Tricus bacterium would look like, covered all over perimeter with flagella. The next thing we're going to talk about is spores. Now, spores aren't real structures of a bacterium, they're more of a lifestyle, okay, a life form. So we'll say a bacteria forms a spore or an endospore. That means it's entering the lifestyle of being in hibernation. Hibernation. And here's an example again where the type of cell wall controls things. Only certain types of gram positives can form spores. Examples especially are the genres Bacillus and Clostridium. Now, gram negatives, because they have a different type of cell wall, they can't form spores. So in a test, if I say you got a spore former, you got to think, oh, it's definitely gram positive. Now you're like, well, what's the purpose? What's the use? Well, when the bacterium enters a spore form, when it goes to a spore form, basically it's protected, but it's asleep. It's put on many layers or coats that'll help it survive things like heating or cooling or starvation or other things. So in this drawing, here's an electron micrograph, and all you're looking at here, the CW, that's where the cell wall is originally. But now it's a spore, and look at all the stuff that's been added. Don't worry what CX, EX, SX stand. Don't worry about that. But a lot of stuff's been put on the outside of the cell wall now to give those. So it's like many layers of coats. So you can just imagine this gentleman here is wearing a coat, probably a coat beneath that. Same way, when these bacteria form spores, they put on many coats for protection, for strength, for to be tough. Okay, think coats of iron even. Rawr. What's the process like going from a normal cell to a spore for gram positives? So you start with originally a happy cell. It's vegetative. It's happy. So we say it's a vegetative cell. The nutrients get exhausted. It's running out of food. There's some kind of stress, temperature. It says, okay, if I don't form a spore, I'm going to die. So it says I'm going to go into the spore state. To do that, it creates a lot of peptidoglycan and lots of protein coats, and it adds those to the surface. All right. So here's our picture here, vegetative, and it's going, and look here, we're kind of showing this, boom, boom. Okay, it's developing that coat of spores. And notice it's just one cell. It's 1-to-1. So if you're thinking about this, okay, well, I'm forming this coat, these coats, and some of these coats are peptidoglycan. Some of these coats are peptidoglycan. So what must it need to transport the building blocks of peptidoglycan? It's got to have more, yeah, bactoprenol. So that's what you'll see in bacteria that are sporulating. They have a lot more bactoprenol than normal because they got to ship out more NAM and NAG to build more of that peptidoglycan to be more protection. Now, okay, but what happens inside? Well, inside, the bacteria's got to get rid of its water. So it's lowering its metabolism, but it's got to really, why? Why does it want to get rid of water? Because if it's going to go through a lot of high temperatures or low temperatures, water is volatile. When it's really cold, it turns to solid ice. When it's really hot, it turns to a gas, and that'll just destroy the cell. So in preparation, it's got to get rid of the water, okay, because of these temperature changes that could be coming. So now what a bacterium does is it basically gets rid of the water and it creates this stuff called dipicalinic acid. And you're like, why is that so important? Well, think about what water does, okay? If you take a cell and you have a protein from the cell, and here's your protein, and there's the hydrophobic parts of this protein. Do they interact with water like this? Or does because of the water, do the hydrophobic kind of hide from the water and go to the center where they're kept away from the water? Yep, this is the correct one. Well, water basically is dictating how this protein folds. And how the protein folds dictates what? Its function. So normally the vegetative cell, if you think about it, needs water to fold correctly. So think about it this way. H2O fold correctly. So the proteins fold correctly and therefore have function. Well, if the water is gone, okay, you'd still want the proteins to stay folded correctly so they can function. So what kind of does the same thing as water in keeping things folded? Dipiclinic acid. It replaces the water so that the proteins stay folded. It prevents, what would be the word? Unfolding, denaturation of proteins, unfolding, denaturation of proteins. So you can see it's very important, right? Dipiclinic is very important. So this whole sporulation process is not simple, but they'll go through it because it can allow for survival. So you're thinking then, okay, got your bacterium sporulated. It's formed a spore now. This cartoon, which was made by a former micro student, is going to explain to you why in the next slide it's so much of a big deal to be a spore. So let's go to the next slide. So then, what's so great about spores? All right, as the little comic strip kind of suggested. Well, first of all, spores make the bacterium very tough and hardy. Now, a spore condition is kind of temporary because if good conditions return, for instance, now there's plenty of food, water, good temperature, everything right, the spore will become a vegetative cell again. It'll shift back from one type to the other. So the vegetative cell is going to be able to grow and reproduce and have a normal metabolism. We say that when a spore goes back to the vegetative cell, it germinates. So you know that term, germination, right? Well, what was so great about spores? All right. Now, depending on the species, a spore can survive some real extremes, things that would kill a normal vegetative cell. So, depending again on the species, some spores are tougher than the others. These are certain things a spore can survive. For instance, a spore can survive. Some spores survive boiling water for 2 hours. Some survive exposure to ethanol. So, for instance, when you're in the lab, and you're spreading stuff, remember, you're working with bacteria, there could be some spores there. So you dip in alcohol and you just don't wait for it to evaporate because some spores can survive alcohol. So what do? You flame it because that flaming, that'll kill the spores for sure. The ethanol, not necessarily, because some spores are tough. Radiation, some spores can survive 1,000 times radiation that a human can survive. Pretty impressive. And some of them are thousands of years of time. For instance, they found some spores that they take out of a mummy, mummies in Egypt. They're thousands of years old. And, well, the mummy ain't coming back to life. But the spore, you put it in nutrient media, right temperature, everything happy, and it will germinate and it'll become a vegetative cell again. So spores are pretty incredible. Well, that ends the story of spores. And compared to spores, I guess you're really not that tough, huh? Okay, well, thanks for listening. Take care. See you next lecture. Bye-bye. To a question. And then here's another kind of a recap question. So Dr. Anticipation, bet you're waiting for that one, is studying a species of Lophotrichus bacteria that form a biofilm and releases endotoxin. What should she expect? So kind of here, what you first want to do is just kind of think, what do the words mean before you get into doing the question. So Lophotrichus, we're talking flagella, a tuft of flagella at one end, okay? Biofilm, well, if thing forms a biofilm, that means it has to have some kind of glycocalyx and or pili. And if it releases endotoxin, it must be gram Negative, right? Okay. So kind of thinking about these things, it's just defining these terms. What's true? It may have a glycocalyces. Yeah, that's true. Possibly, yes. It cannot sporulate. Yeah, that's correct also. If it's gram negative, it can't sporulate. It has flagella. Yep, Lophotrichus means it has flagella. Trichos, flagella, yep. So the answer has got to be E, all of the above. All right, great. Hope that makes sense. So let's start a new topic, microbial growth, reproduction and growth, and with nutrition thrown in there just for good. And we're going to mainly focus on bacteria here. So bacteria reproduce asexually by binary fission. Fission, you know, things splitting, binary two. So when you say a binary fission in bacteria, you want to think the result is two identical daughter cells, and they are identical to each other in their DNA, and they're identical to the mother in terms of their DNA, unless there's some mutation in the DNA, and we'll worry about that later. But right now, assume the DNA is identical between the daughters' cells. The steps in binary fission are the next page. I just have a little picture here taken with a microscope, and you can see the end of binary fission. You have two, okay, look at them, you have two daughter cells here, and they look pretty much identically the same. See right there, one, and there. And you can see the splitting. All right, so what happened here was fission. All right, and each of these is a daughter cell. Here's the steps. So what's not shown here is the chromosome has already replicated. So the chromosome is replicated already. So now you have two chromosomes. And remember, one of the jobs of the plasma membrane is to anchor the new chromosomes for each daughter. So you could say here's a hook or an anchor site here. Here's a hook or an anchor site there. So they're kind of separated, so when the cell division happens, the invagination happens here, one is gone to one chromosome goes to the other, and then the cross wall forms and they separate. And the result, as you can see. Now, usually the daughter cells completely separate, they detach, but sometimes they don't. They end up kind of staying around, sticking with each other. If they don't, that's what leads to the arrangement we've seen: strepto, klepto, a staphylo, diplococcus, etc. It's kind of like an incomplete fission. So here we have some streptobacilli, and you could imagine they went through fission. You might have had one, it divided into two, and then those two divided again to four, right here, shown here, four daughters, okay? First division, and then from the second division, but they're all still stuck together, so that's why they're in a chain, streptobacilli. Now, if you're trying to count the number of cells, all right, you think, okay, one goes to two divide now, and now you get four. So we're kind of going, four divide, and you have eight, and so forth. Okay, you got all these things going on. And so you think, wow, bacteria grow fast. Yeah, they do. And in this example here, they're dividing every 20 minutes, right? Every 20 minutes, they're doubled. Looking at these numbers here, right? You see that? Okay. So this would be true if they were continuously growing by fission and not slowing down. It's an exponential growth. All right. Now, if you're looking at this and you say, well, they've doubled every 20 minutes, we'd say now the generation time in this example here is 20 minutes. All right. That's the word generation time. It's the time required for the population to double a number. And this happens, but only through certain periods of growth of a population of bacteria. It's really not like this happening all the time. Okay. So there's actually 4 phases of growth. So if you're taking bacteria and you're growing them in a flask in a lab, or if somehow they've infected some tissue, maybe a cut in your arm, they're going to double. There's going to be a one period where they double like this exponentially, but they're not going to do it always because the environment controls kind of what's happening. That's the key you want to think about. The environment controls what's happening in these four growth phases. So you'll see this graph quite a bit. On one side here, the Y, we've got the log of viable cells. Remember, viable is living cells. We're only looking at living cells here. Living. Not dead and living, just living. And we're taking the log 10. So 10 to the 2, here's 100. 10 to the 3, here's 1,000. Okay? 10 to the 4, here's 10,000. You get the number. Okay? It's a log scale. And here's time. All right, so 100 to 1,000 to 10,000, okay? So the first, let's say you put bacteria and they go in a fresh medium, okay? Or a fresh wound. And you'll see a lag phase first, right here. And then after a while, their growth rate really picks up and we have a log phase next. And notice, the bright red are the living cells. Don't forget that. And the really dark are the dead cells. So you can see during log, wow, there's a lot of living, not many dead. And then we go into the next phase, we go stationary phase. All right. And you look at in terms of living cell concentration, look, it's not changing, right? That's the thing people forget. In stationary phase, the concentration of living cells is pretty much staying the same. But over time, you see there's a lot more dead ones now compared to living than before. Okay, comparing these two. And finally, with enough time, you go into death or decline phase, the bacteria. And here you're saying that the concentration of living ones are going down now. In death and decline, the concentration of living ones are going down. So let's kind of, again, focusing on viable cells here, let's kind of just explain each of these again. You can kind of jump back. Can't fit it all in one slide. So let's look at these and let's kind of write it out. What's going on here? You have bacteria, you put them in fresh glycerol-based medium. All right. Well, at first there's lag, and lag is very slow. You didn't see the change in number of living cells was pretty small because the bacteria are adapting to the new environment. Maybe they came out of some glucose or something else, and we've put them on glycerol. So they're adapting to the glycerol. They have to make enzymes to adapt, and that kind of slows down their growth. Okay, they have to adjust. Then they start taking off. Okay, the concentration of living cells really starts going, and it's truly exponential like we saw in that first chart. 2 to 4 to 8 to 16 to 32. This is log or exponential. They double at a constant rate every 20 minutes. The reproductive rate, birth rate, is much greater than the death rate. Then the flattening, the stationary. In stationary, the reproductive rate equals the death rate. Well, why? Because of the environment. There's not enough food around. Waste products are building up. So in this case, you'd say over stationary phase, the total concentration, living and dead, that's increasing. Because there are births, but there are deaths, but there are births, and total living and dead is increasing. But the living concentration is staying the same, right? It's just there's more and more dead ones. So that's why the total is increasing. And then finally, when you get to death and decline phase, the concentration dying exceeds the number that are being born. So the death rate is higher than the birth rate. So overall, the concentration of living cells, the viable cell concentration, goes down. Now there's still some fission happening, but the living concentration is going down because the death rate is bigger than the birth rate. If you have gram positives, okay, environment really gets tough in stationary and in death. What are you going to start to see when gram positives are going through stationary and death and decline phase? What do you think you'd see if you looked under a microscope? What would you see if you had a really strong microscope? You'd see the formation of spores. Very good. Spores would start to form. All right. Stationary and death decline phase in gram positives. This is just another take on it. Okay, it comes from the OpenStax book, so hey, we have to pay tribute to them. Okay, they're free. Come on. And again, lag phase, no increase. It's a small increase as they adjust. And here, the living cell, the log phase, really goes up big time. Exponential increase. Stationary phase, rate of cell division, rate of death, same. So it plateaus. Concentration of living cells, we're looking at concentration of living cells. And then death decline, concentration of living cells is going down. All right. Real important to understand that. Now, something I want to remind people, and people sometimes forget this, it seems to be obvious, but it's often overlooked. If you look at a colony on a plate, okay, a colony on a plate has millions to billions of cells. All right, remember Cook. He's using a flame loop. He streaks out the cells and he puts one here. And that one cell, so we're divides many times into a colony. So if you looked at under a really strong microscope, here's the colony. Maybe this one right here, this is where the first cell started. Okay, after it got streaked out. And then it divided and divided, and it went through lag and log and stationary, and maybe it's an old plate, so now it's in decline. And that one divided and divided, and now what do you got? You have all of these colonies. Sorry, all of these cells in this colony. This is one colony, but it started from one original cell that went through those four phases. Okay, which is pretty amazing when you think about it. So you're doing single streaking, you streak out, you get one here, one here, one here, and with time goes through the four phases, and now you have millions, but they all came from that one original cell that was streaked out. Kind of cool. Now, let's talk about in growth. We're going to see how certain things affect growth. The first thing we're going to talk about is temperature. Okay. Now, different bacteria grow at different ideal temperatures. All right. So if you have bacteria that are called psychrophiles, for instance, and you got to know these definitions, these grow ideally between 0 and 20 degrees Celsius. So that's basically from really cold ice, really cold ice to about a little bit below room temperature. Mesophiles are next. They're the ones that grow around room temperature to body temperature, 20 degrees Celsius to 40 degrees Celsius. Thermophiles are bacteria that grow from above body temperature, 40 degrees, to close to boiling, 40 to 80 degrees Celsius. And hyperthermophiles are the ones that grow around boiling, even above boiling. Okay, I knew a scientist and he studied hyperthermophiles, and he literally grew these things in crock pots, and they were literally growing in boiling nutrient media in these crock pots, and they were on 24 hours a day. So you can use crock pots for a lot of things, folks. Now, what you want to think about is what's true of these temperature ranges. Well, these temperature ranges, the bacteria grow here because this is where their proteins are active. This is the temperature of their proteins. So they live, they survive because that's where their proteins are active. So you say, if I take a hyperthermophile, okay, for instance, if I take this guy, all right, and I took him and I put him at, room temperature, his proteins would not be active. He probably would be dead because his proteins would be sitting there doing nothing. But at really high temperature, that's when they're active. That's when they're moving. That's when they're doing their job. That's kind of a hard idea to get your mind around, but that's how it works. Okay? So let's kind of get into a little more specifics here. If you say I've got an obligate or strict thermophile or a mesophile, that means this range here is not just ideal. This is the range that they live at only. Only at this range. So if you say I've got a strict psychrophile, that means if you go above 20 degrees, it's dead. Okay. If you have an obligate, let's use the fancier, obligate thermophile, that means, okay, 40, 50, 60, 70, 80, yeah, it's growing great. But if you go below it or above it, it's dead. Okay. Now you get into the tolerant or the tropic ones. So, for instance, if you say a mesophile and you say it's psychrotropic or psychrotolerant, this one, because we know what tolerant means, we use that more often than not. Well, that means you've got a mesophile and it wants to grow 20 to 40. It likes growing 20 to 40, but it can grow maybe around the fridge. Maybe it can grow in the fridge like 4 degrees Celsius or 10 degrees Celsius. So a psychrotolerant mesophile, you'd think, okay, ideally, if it has its druthers, yeah, 20 to 40, that's when it grows best, somewhere between 20 to 40. But it'll grow lower temperatures even in the fridge. And that's why, folks, like the psychrotolerant mesophiles, they'll grow great when they're outside the fridge. They grow really fast. But if you put them in the fridge, they'll still grow, all right, in the fridge. And that's why your food will spoil in the fridge, because these spoilage microbes, yeah, they're mesophiles. They grow really good at around room temperature. But even if you stick them in the fridge, they're still going to grow a little bit. So they're still reproducing there. So that's why you've got a psychrotolerant mesophile, right? Again, I like to always show some practical applications of what we're learning here. All right. So now let's talk about oxygen. And again, we're going to look at terms that we see in the Berge's manual. If you have a strict or obligate aerobe, that means it must have oxygen to grow. If you have a strict or obligate anaerobe, that means it cannot tolerate oxygen. If there's oxygen around, it dies. Must live in an oxygen-free environment. Now, facultative is basically means it can grow in the presence or absence of oxygen. It's like the great guest that comes to your house that's really flexible. Is it, if it's really hot or really cold, they're happy. All right, they can adjust, it seems like it. They're very flexible. If a bacteria is facultative, it can grow in the presence or absence of oxygen. If there's a little oxygen, a lot of oxygen, it's fine. Facultative. Sometimes those are called aerotolerant. We won't worry about that. Then we also have microaerophile. Now, microaerophile is the bad guest. That's the person that comes to you that needs everything within a certain range. Very, very picky. All right. So if a microaerophile, it needs low oxygen to grow. But if there's too much, it dies. If there's too little, it dies. Kind of reminds you of Goldilocks, okay? The original bad guest. Remember Goldilocks? Not only is she a thief, okay? Not only is this young lady a thief, she's a picky thief. She'd be kind of a microaerophile if she were a bacterium. Because, if the porridge is too hot, she don't eat it. Porridge is too cold, she doesn't eat it. It's got to be just right. Microaerophiles. If the oxygen's too much or too little, not good. And you've got some microaerophiles in your gut. That's just an example you can think about. They have to only a certain range, they'll grow. Okay, and oxygen. The difficult, demanding guest. This picture kind of sums it up. So you've got a tube here. You have a tube here. And you could imagine there's oxygen up here. Oxygen is up here. Okay? So an obligate aerobe is going to grow very near the tube where there's a lot of oxygen. See, the cells are growing near the oxygen. Facultative or aerotolerant, just worry about it. They can take or leave it. They grow where there's a lot of oxygen or a little oxygen. They're fine. A strict anaerobe is growing far away as possible from the oxygen. Okay? Oxygen will kill it. And finally, your microaerophile, look, if there's too little oxygen, it's dead. If there's too high of oxygen, too much, it's dead. It can only grow in this little range of oxygen percentage, microaerophiles. Okay, if you're a lab scientist, it's much easier to take the first three. Really, the fourth one, the microaerophile, is a pain. Okay, you can have plenty of oxygen. You can even create an environment where there's no oxygen, but always keeping it in a certain range, a very tight little range, because usually the bacteria are using it. So they're going to use some, which means it's going to go down. So you got to add it back, but you can't add too much. So microaerophiles, very difficult to work with. So we've done temp, we've done oxygen. How about pH? So most bacteria, have a range of, you'll find some between pH 2 and 9.5. Okay, you'll find bacteria in all these pH ranges. Now, why is pH important? Because just like temperature affects protein function, pH affects protein function. Okay? If you go think about Bio 61 and you have like amino acids, and you think, let's say you have an amino acid and here's an N plus, okay, and here's an O minus. Oh, why did that line show up? That wasn't supposed to show up. You have an O minus, and okay, there, but if suddenly there's a lot of H pluses around, and that H comes here, well, that ionic attraction's kind of diminished, right? So the H plus concentration, which you want to think of, the H plus concentration, it affects protein structure. Okay? And again, the proteins, if you change their structure, you're going to change their function, their activity. So pH affects protein structure, and that'll affect activity. Therefore, they're very sensitive to pH. Most bacteria like a pH around neutral. For instance, the human blood is neutral. A lot of bacteria grow there. So we consider those bacteria neutrophiles. Oh, geez, forgot the E, sorry. Neutrophiles. Neutrophiles, sorry. these mean their proteins are active around pH 7. Neutrophiles. All right, so most bacteria are neutrophiles. Now, therefore, most bacteria do not grow well in acid, except, and we got to talk about these, the acidophiles. These are acid tolerant bacteria. They like to grow in acid, and they're useful because they grow in acid. They're useful a lot in the food and dairy industry. Think of acidic dairy products like yogurt, some cheeses. They're acidic, they're tart. Why? Because these bacteria that are growing in them are acidophiles. So acidophiles are used because they produce the acid in metabolism. And they produce it, they create it in their environment, and they tolerate it. So they produce it, and they tolerate what the acid they produce. Now, why do they do that? Because it's a dog-eat-dog world. It's a competitive world. And most of the bacteria that are in, for instance, milk are neutrophiles. But if these bacteria, the acidophiles, create a lot of acid, the neutrophiles can't grow. So this allows them to outcompete the neutrophiles. And now these neutrophiles, they're the ones that usually cause spoilage. So if the acidophiles make acid to stop the neutrophiles from growing, and these neutrophiles cause spoilage, well, the acidophiles therefore are going to prevent spoilage. So we say yogurt is tart. Yeah, it's tart, but it prevents the spoilage of milk. Okay? Yogurt is just milk where these acidophiles have made a lot of acid, and it preserves it, and it stops the neutrophiles, the spoilage bacteria, from growing and making the milk turn bad and smelly and spoiling it. So when you think of these acidophiles, they like that environment. Their proteins are active at acidic pHs, pH 3 to 6. That's when they're most active. Okay. Here just sums it up. An acidophile, you can see its proteins, it grows best. Its proteins are active around pH 3. Okay. Neutrophiles around pH 7. That's when their proteins are active. That's their environment. And there are even alkalophiles. Alkalophiles grow at alkaline pH, around pH 9.5 or 10. There are some foods that are made with these alkalophiles, and basically they have a kind of a weird taste because they're making base. They're growing in a basic solution. So it's kind of a weird kind of thing, some of these things. All right, so this kind of, now again, most bacteria are neutrophiles. They grow best around neutral pH. Acidophiles have optimal growth around 3, alkalophiles around 9. Okay. So let's look in these things here. Where does the acid come from? Well, yogurt, like I said, acidophilic bacteria make acid. That gives it the tart thing. If you're from Vietnamese background, you might have seen these. These are like raw spiced meat. And they're not refrigerated. They're called nem chua. And they're tart, and they're tasty, and they don't make you sick. Well, basically you've got the meat and the spices, and you put acidophilic bacteria that make the acid. The acid stops the spoilage neutrophiles from growing, and that's why it's safe to eat. Same thing with salami, which is Italian in Spanish. It's raw meat with spices. They put acidophiles. The acidophiles make acid. They stop the growth of neutrophils. Neutrophils cause the problems. They cause spoilage. Sometimes they even make poisons that cause death. Kimchi. Kimchi, if you're from a Korean background, same idea. Acidophilic bacteria make acids. Acids preserve it, stop the neutrophils from growing, stop the spoilage microbes from growing. All right. So they're very useful. Now, what's kind of interesting, people who do like food history, they're like, well, why did these come to be? Was it that people kind of discovered that you put acidophiles in here and it gave things a tart flavor? And just by accident, hey, not only did it give it a tart flavor, but it preserved it and stopped them from spoiling? Or was it the other way around, that they noticed originally that if you put acidophiles in it, it preserved it, it stopped the spoilage, and basically if you ate it, you didn't get sick and you didn't die. So they realized, hey, these acidophiles are needed to make sure we stay healthy and happy, and they just got used to the flavor after that. Okay, it's not clear why they originally started putting acidophilic bacteria in there. Was it for the flavor or the preservative? But we know it does both.
Alrighty, well, then folks, that's it for the day. And next lecture, we'll talk about metabolism. Take care. Bye-bye.